Good day. This is the Professor, CBMD PhD, and today is Tuesday, July 18th, 2023. It is 2:53 and 46 seconds, a.m. Eastern Standard Time, and today's research, cell repair. So let's jump to it here. DNA element with a murky past is borrowing. Cells repair machinery. Cellular DNA, thought to be an accidental byproduct, is borrowing the cell's DNA, repair mechanisms, to copy itself. Date July 12, 2023. Source, Duke University. Like their viral cousins, retrotransposons have been found borrowing the cell's own machinery to achieve their goals. They hijack a little known piece of the cell's DNA, repair function, to close themselves into a ring-like shape, and then create a matching double strand. The finding happens 40 years of conventional wisdom and may offer new insights into cancer, viral infections, and immune responses. It can also offer a new way to insert sequences into the genome. Retrotransposons are segments of DNA around 7,000 letters long that copy and paste themselves into different parts of the genomes of both plants and animals. And by doing this, they play a role in rewriting DNA and regulating how the cell uses its genes. Retrotransposons are believed to be behind a lot of the variation and innovation in genes that drives evolution and are inherited from both parents. Many studies have suggested that these rings of DNA outside the chromosomes are somehow involved in the development and progression of cancer, in part because they are known to harbor cancer-driving oncogenes within their DNA sequences. The retrovirus HIV, which causes AIDS, is also known to form circular DNA. The scientific investigators and researchers think these elements are the source of genome dynamics for animal evolution and even to affect our daily lives. Retrotransposons are quite common. They make up about 40% of the human genome and more than 75% of the maize genome. But how and where they copy themselves has always been a bit murky. The researchers hold up a thick textbook on retroviruses that they consulted for this study. The book says the ring-like sequences are created by recombining the two ends of linear DNA that are just a dead end, a byproduct of failed replication. In earlier work with fruit fly eggs, the team had established that inherited retrotransposons use in their cells, which support the egg, as factories to manufacture many copies of themselves that are then distributed throughout the genome in the fly developing egg. The model system allowed the researchers to zoom in still further to learn more about retrotransposons. In the latest work, they found unexpectedly that most newly added retrotransposons were in the circular form rather than being integrated into the host genome. They then ran a series of experiments, knocking out the cell's DNA repair mechanisms one at a time to figure out how and where the circles are being formed. The answer, a little studied DNA repair mechanism called alternative end joining DNA repair, or LDJ for short, which repairs double-stranded breaks. The retrotransposon sequences were using this part of the host repair machinery to sew the ends of their single-stranded DNA together, and then using its DNA synthase to create a matching double strand for good measure. The researchers confirmed that this is also the process within human cells. And so retrotransposons aren't a sloppy accident. They're actually hijacking a little bit of the cell's machinery to manufacture more of themselves, just like viruses do. The discovery actually overturns the textbook model. They show that the recombination event proposed by the textbook is not important to forming rings. Instead, it is like the LTJ pathway, driving circular production. The laboratory is currently trying to test whether circular DNA can be intermediate to make new genome insertions. They are also testing whether circular DNA can be sensed by our immune system to trigger an immune response. In the retro field, the retro transposon field, people think circular DNA is just a minor event, but the study is bringing circular DNA into the center stage. People should pay more attention to circular DNA. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.